Good morning, everyone. It's my pleasure to tell you a little bit about Opus Genetics this morning. So Opus is a really unique company that's focused on patients with rare inherited retinal diseases. You know, in rare disease, you get to know the patients, the kids, and the families. And we decided to start a company because we saw a problem. We noticed that there were a number of really promising gene therapies in academia that were just not moving forward at the pace that we wanted to see. And it's even great labs like Jean Bennett's lab at Penn. And so, you know, she's been busy in the last 10 years since co-discovering and developing Luxturna. So we decided to start a company that was really focused on fast and efficient retinal gene therapies. That is, it's a no-brainer, right? Capitalize on the path blazed by Spark and the Luxturna program. Develop products that are fit for use. So if we don't need to go back and redo a capsid or something like that, just take it forward if it's good enough as it is. And then manufacture using modern methods, but at a scale that makes sense. So our first few programs are depicted here on our pipeline slide. The first two assets came from Jean Bennett's lab at Penn. The second one came from Eric Pierce's lab at Harvard. And these are coincidentally all LCAs, but that's not the strategy the company will take on any IRD. But you can see here from the chart that we're on pace to have one IND filing per year for the next few years. And we're excited to get our first program in the clinic at the end of this year for LCA5. RDH12 is about a year behind that, and NMNAT1 is about half a year behind RDH12. And I'll tell you more about those in a second. So I'm joined by Ash Jagopal as our Chief Scientific Officer, Joe Shackle as our COO, and Jennifer Hunt as our Chief Development Officer, and then Nikki Heron as our part-time CFO. You can see that there's a lot of relevant experience here for rare disease, ophthalmology, and gene therapy. Our scientific advisory board is chaired by Jean Bennett. She's also on the board of directors and a co-founder of the company. She's joined by Eric Pierce, excuse me, who's also a co-founder, Radha Ayagari at UCSD, and Sanford Boy at University of Florida. And finally, our clinical advisory board, joined by Dan Chung, Elise Heon, Jose Sahel, and Bart Leroy at Ghent. So I want to take a minute to talk about this special relationship with the Foundation Fighting Blindness. And it's not just because the foundation incepted and lead financed the company, but it's more about the relationship going forward. That is, we can work with the My Retina Tracker registry to help pinpoint prevalence. We can work with the clinical consortium to help us optimize our trials and our endpoints, access to patients. We can even collaborate on natural history studies. Essentially, we want to become the partner of choice of the foundation to develop emerging therapies coming out of their granting portfolio. We have a great partnership with Resilience. This is a new CDMO that's really technology focused. They've raised over $1 billion themselves. And coincidentally, they bought the Bluebird facility in Research Triangle Park, which is only about 2 miles from our lab. So it's really great to be working with them, and it'll be easy for us to have a person in plant during our campaign runs. Also working with Thermo for ourselves and Dark Horse Consulting to augment our CMC team. So let's talk about some of the programs here. LCA5 is an early onset severe retinal degeneration. It codes for the labracillin protein. This is a protein in the connecting cilium of the photoreceptor that traffics proteins up and down, and when that's dysfunctional, it leads to profound vision loss early in life. The important thing here is that these patients have structural functional dissociation. That is, they retain enough retinal structure into their second and third decades of life so that we can go in with gene augmentation and potentially restore that function. Some really important work was published by Jean Bennett's lab showing that if you go into a faithful animal model and you replace the broken gene, as long as there's 30% or more of the ONL remaining, we can actually restore structure and function. And we tracked some patients in natural history study, the red dots you see on the figure on the left. We have many patients that are 50% or more of ONL layer remaining. Really cool results on pupillary light response as sort of an early biomarker of potential efficacy. Lots more data in the paper, but the figure in the middle, you see it's a flat line on pupillary light response, wild type on the left with a full response, and then rescue with gene therapy and the knockout on the right completely restores that eye-brain connection. The second program is RDH12. This is an enzyme in the visual cycle, but unlike Luxturna, it's actually in the photoreceptors, not the RPE. This condition also has structural functional dissociation, and we're excited to bring this into the clinic around this time next year. Our third program is also an enzyme target. This one is involved in NAD biosynthesis, so it's involved with the energetics of the cells. So you know that if you have any problems there, the retina goes haywire. Another great animal model showing that we can restore structure and function in this severe IRD, and we'll be in the clinic in early 2024. So real quick, at a high level, the company, we raised a $19 million seed round. That gets us into early next year. We're actively raising an A round, which will bring in several important clinical results on multiple programs. And I'll leave you with this bubble chart. It just tells you about the world of opportunity here. It's a target-rich environment, and with the toolkit we have now, we can tackle many of these, and as the toolkit improves, we'll be able to go after most of these. So thanks a lot for your attention.